17, looking at meiosis, function, and sexual reproduction.
And so, in the interest of time and also not to go over a material that you already know, I'm going to just go through this very briefly, not in terms of content, but just how you could potentially use this as a way to communicate information with your students. So we will be making this presentation and the related links available to you on the website not tomorrow, but it will be available to you. And then I'll be showing you an activity that you could do using manipulatives that you could potentially help your students retain the information about some relatively complicated subcellular processes. Okay, so the first thing I'll start with your bell work. So while your students are getting ready for the day, at this point, before starting to talk about meiosis, you would have already had the prior knowledge about mitosis and how mitosis works within the cell to create somatic growth. So first off, you want to think about how organisms are growing. And so some organisms have asexual growth. So this is a polyp of a cnidarian, the phylum Cnidarian. Those organisms grow via asexual reproduction. So just mitotic growth allows the formation of their offspring, which is these buds that grow off of the parent. And so if we were to have mitotic reproduction and we were just to be able to produce our offspring just through mitosis, we could look like something like this. But in truth, we don't reproduce like that. Instead, we have a gametic life cycle in which we have gametes that are produced through the process of meiosis. So something to engage your students a little bit. And then you could go through some of your content. Okay, so meiosis, we're going to talk about how meiosis functions and that it is extraordinarily useful in terms of sexual reproduction. And so not only are we going to be talking about 1617, but also this is going to cause a review of 1614, talking about the process of mitosis and describing the process of meiosis as well. Okay, so one of your hooks, of course, everybody loves a chance to talk about sex in the classroom, right? Because you're talking about high school students that just can't get their minds off of sex. So we'll be talking about sex, and then at the end, we're going to be doing it on the table. So your students will be doing their mitosis and meiosis on the table. All right, so please don't feel like you have to use your clickers with this one, but this is one way that you can start using clickers in your class. Make sure that they are all in their seats. They're getting ready for the day. So starting to ask them questions about just some simple information. So which of these is transmitting genes from one generation to another? DNA, gametes, somatic cells, and then through this, I put PowerPoint. So you can do this, show them the answer. So of course, your gametes are being useful for transmitting the genes from one generation to the next. So in order to start here, we need to review about sexual reproduction. So with our sexual reproduction, we're talking about organisms. They're going to reproduce with gametes. Gametes are different than your sex cells, and you can talk about the difference between diploid and haploid types of cells. With your sexual reproduction, you start off with your male. In humans, you have your male gametes, which are your sperm, your female gametes, which are your eggs. And if you have fusion of your two sex cells, you end up with this fertilized egg, which is your zygote. And of course, on one mechanism, you could help your students with taking notes, either using your Cornell notes. So what is highlighted is information that your students could write down. Or you could also provide them with their blank sheets. And so in your folders also, you'll notice that for this, everything that's on the right-hand side is related to this lesson plan. So you'll see that there is a lesson plan guide related to this and a packet of materials that we're going to be going through in addition with teacher directions and handouts for students. And I recognize that this is stuff that you guys have done previously, but if it's something you can start thinking about maybe including in your lesson plans for the coming year, it's a possibility. All right, so the reason we're talking about this, of course, is that when we're talking about sexual reproduction, it is essential that we understand meiosis. And it is meiosis that's going to produce us with your gametes. And humans, of course, are eggs and sperm. And the end result of meiosis is that we're going to end up with these 4 daughter cells. And these 4 daughter cells are different than their parent cell in that they have half the amount of genetic material as the parent cell. And so with this, it of course occurs in your testes or your ovaries, depending on the person creating them. You can go over chromosome numbers, and of course this will allow you to be able to talk about the difference between chromosomes and sister chromatids, and to differentiate that. So in this figure, this is your karyotype of a human genome. These are created through the use of somatic cells in the process of mitosis. And so you have your 23 pairs of homologous chromosomes. So this is one pair of homologous chromosomes, another pair. And within your homologous chromosomes, you have your sister chromatids. Okay, so here's your karyotype of your human genome. So total of 46 chromosomes. Since it's diploid, 2n equals 46, and you have 23 individual pairs of homologous chromosomes. So if you're looking at your homologous chromosomes, it consists of these 2 individual sister chromatids attached with a centromere. One of these you get from your mom, and of course one of them you get from your dad. And even though these two chromosomes, they each are twins in that they carry the same type of material, they carry the same types of genes, so they can code for things like hair color or eye color or height, but you can have a different variant of that same gene. So you might have one gene that codes for eye color, and it occurs on both of those sister chromatids. Both of those homologous chromosomes, but on one of those it could code for the color brown, but on the other it could code for the color blue. And so this variation can be seen within the resulting organism. And so with these, you can give specific definitions of homologous chromosomes or sister chromatids. This is a common misconception among your students. I'm sure you are familiar. It's still a common misconception when you get to the college level, too. So the more times they hear it, the better off they're going to be. And so with your sister chromatids, they're 2 copies of a single chromosome. They're attached to each other. And we have pictures of these to reiterate. So we're presenting this material in multiple representations to reiterate to your students. And after this type of material, you could even have your students, you could ask another clicker question where your students were, where you asked your students questions about a figure of your sister chromatid versus your homologous chromosomes, or you could potentially ask students to pair up and draw the different things, or show them pictures of the chromosomes and ask questions about it. Okay, so you talk about your sex chromosomes and how within our humans we've got these 23 pairs. And of course, there is a difference in your females and your males. So with your females, you end up with, on your 23rd pair of homologous chromosomes, you end up with 2 X's, but your males have an X and a Y. And so this pair is not identical. So with the females, these 2 X's that females have, they are homologous, but with your males, the X and the Y are very different. The Y, of course, does not contain as many genes as the X, but it does contain some genes that are not on the X chromosome. So if we look at a comparison, Y is much smaller than your X in terms of the number of base pairs, but there are some of these genes that are not present on your X chromosome. And then, of course, it's essential that they understand the difference between diploid and haploid cells when you're talking about meiosis, especially since we're talking about this reduction in genetic material. So we have these 2 sets of chromosomes in our diploid cells, and we have 1 set of chromosomes within our haploid cells. And with this, within our adult form, we have diploid adults, 2 sets of chromosomes, but as we have this process of meiosis, this reduces the amount of that genetic material by half. So our gametes are haploid cells, but the process of fertilization restores us to that diploid state, and then the remainder of our cells undergo mitosis for growth. So talking about our diploid cells, diploid cells are individuals that have 2 sets of chromosomes. So you could look like a diploid individual, even if you are just talking about these individual chromosomes that have a single chromatid, or you could look like a diploid if you have 2 sets of homologous 2 sets of chromosomes that are paired sister chromatids with at their centromere. And of course, there's ways that you could assess whether students understand this by looking at this relationship between the 2n equals 46 versus 2n versus 8, and having students calculate that number themselves. With your haploid cells, you'd want to go through how your haploid cells can have 1 or 2 chromatids, and then asking questions about whether or not they understand if this is haploid or diploid. So, of course, with this is a diploid cell, so they could differentiate that because you have matching chromosomes. And then, of course, asking a reiteration question to see if they followed that. So the reason for being able to answer that previous question is because your cell has multiple sets of chromosomes. All right, and so going over it again, understanding that when you have these different cells, you go from this parent that is diploid, you produce haploid gametes, and then fertilization restores this diploid state to make your embryo, and talking about the process of fertilization. And of course, going back to this idea of the zygote and mentioning where in this life cycle you have these different processes. Diploid adults, meiosis, haploid gametes, fertilization, restores diploidy, then mitosis. And part of the reason that we have importance of meiosis and why it's so essential is that within our genome, we have 46 chromosomes in our eggs. We have these, if we had 46 chromosomes in our egg, and if we had 46 chromosomes within our sperm, we would end up with an individual that had 92 chromosomes. And if it was necessary for us to have an organism with 92 chromosomes mate with another organism that had 92 chromosomes, you'd end up with an organism that had 184 chromosomes, and it would be a never-ending process, and that would be a little excessive. And it would be extraordinarily rare for an individual with 92 chromosomes to find another individual with 92 chromosomes. So, meiosis and mitosis. Okay, so then going through the process of meiosis, you would want to walk through, I'm not going to walk through all of the process of meiosis with you. I want to get us to some group work. But with meiosis, you would walk your students through the different stages. What I'm going to do is just get you show us our video clip of meiosis.
Produce gametes, eggs, and sperm through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair. resulting in a tug of war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
And that's the end.